It's with great pleasure I invite our next speaker, Professor Diane Simeone, who's the Laura and Isaac Perlmutter Professor of Surgery and Pathology at New York University, to present her surgical keynote, New Platforms to Improve Survival in Pancreatic Cancer. Thanks, Diane.
Thank you very much, and it's really been so special to have a chance to come to this meeting and to share with you some of our work today. So we're going to just talk about pancreatic cancer, where we are and where we hope to go, and I'm going to show you a couple of things that we've built out that we hope may be a path to a different future state. Here on this slide, we have data about the grim facts of where we are with pancreas cancer today, 12% survival rate. Really, as a surgeon, a frustrating disease because only a small percentage of patients actually are amenable to surgery. And then just for everybody in the audience to know who doesn't take care of patients, even if a patient is surgically resectable, it doesn't mean that it's early stage disease. Most of those patients actually have 2 to 3 centimeter tumors and positive lymph nodes. So this really is a picture that hasn't changed that much over the last 30 years. Now, as a surgeon, we have certain tasks at hand, which to make sure we operatively choose the correct patients, to optimize therapeutic approaches, to achieve negative margins, to expand surgical approaches, including vascular resection when appropriate and may provide benefit to patients, and to identify and resect high-risk lesions. However, beyond that, we have a lot of therapeutic challenges with pancreas cancer, which really focus around the difficult biology, and I list some of the known challenges of the biology in this slide. Just want to talk about what we know about risk. We know there can be a genetic predisposition. We'll talk about that a little bit more further in the talk. But even of the modifiable risk factors that are listed on the slide, these all increase the risk only about twofold. And I would say as a clinician who sees patients with a new pancreas cancer in the clinic, about half of patients I really still can't put my finger on why they got the disease. So there's still a lot to uncover. We know that blacks are at increased risk of getting pancreas cancer compared to whites, and there is an age prevalence, although we are seeing some increase in pancreatic cancer in people in younger ages. So this is actually a slide I made in 1995 when I finished my residency, which is if surgery is often an option in only a small number of patients, what we can do to try to make a difference in this disease. And really for the first part of my career, I worked on basic science. I continue to have a basic science laboratory and focused on this first part, which is to better understand the molecular mechanisms of pancreatic cancer development and progression. But what I'm going to talk about today is really trying to tackle these two latter points in a different kind of way, which is develop more effective therapeutics and then detecting pancreatic cancer early. So we have had some advances. In fact, I was just at the AACR pancreatic cancer meeting in Boston in September, 700 researchers, and if you open up any major journal, you'll find a very interesting paper about pancreatic cancer biology, so the scientific community has grown. We now know that not all pancreatic cancers are the same, that they're heterogeneous in their nature, and some of them have different molecular signatures. And of course, along with this, we have a different toolbox with therapeutic agents to try to tackle it. There are lots of areas of active research that are important. A number of these we and others are working on. But the key question is, how do we take this and then have it make a difference for patients? And as I found some of my own work going into the clinical trial space, I realized there were unique challenges there. And here I just say, for all these different therapeutics that are out there, how do we go about doing this in a logical way that's efficient for patients? Now, on the clinical trial side, there are actually a lot of barriers for clinical trial success in pancreatic cancer. First of all, it's hard to move compelling basic science into clinical trial arena. It's particularly hard for pancreatic cancer, where there have been a lot of failed trials, so there's reticence of drug companies to move into this space. From an academic standpoint, we don't do a very good job of data sharing. We don't have good predictive biomarkers, and we haven't really studied what's going on in patient tumors as we treat them. If you add into that only 4% of patients with pancreatic cancer get on clinical trials, you can see that this is not a recipe for success to make a difference. So basically, I would say a couple of years ago, I had an epiphany that I was going to have my entire career go by and we were not going to move the needle on pancreas cancer. And so we decided to entertain a new strategy. And I actually approached the Pancreatic Cancer Action Network, a lot of recurring themes about the value of advocacy organizations, and said, can we rethink how we do clinical trials for pancreatic cancer? And of course, they were a very well-regarded organization, and they actually served as the fabric and glue to do this and made a significant investment to help us develop a new clinical trial ecosystem in the United States, which we have called Precision Promise. And this was really an interesting project where we really started in a room with about 8 or 10 people with a piece of white paper and said, how should we do this? And we started off just saying, these are the basic tenets that should guide us. We should have a novel model of cooperation. Trials should be rigorously vetted by expert scientific teams. We decided to have an adaptive clinical trial design. I'll show you what I mean by that. We thought we had to enter a new era where we had deep learning from every patient, ongoing sharing of data, flexibility, and of course we couldn't do this unless we had the pharma industry partnering with us and kept keeping everybody honest by putting the patient at the center of every decision. So what we decided on was an adaptive platform trial, and these are starting to roll out, but they're still a relatively new concept. And as you can see on the bottom of the slide, for those who are interested, that's a review we wrote to kind of educate the public about the nuances of adaptive platform trial design. But this is a nimble trial design that allows us to evaluate multiple therapies simultaneously. You have shared controlled arms, so we have fewer patients getting standard of care. These trials are smaller in design, and they can be more effective to evaluate biomarkers. You probably save about a third to half of the time and money to evaluate a therapeutic. We really thought it was important to put in place that every patient got biopsies before and on treatment so we could understand what therapies were doing, why they worked or didn't work, and we really wanted to have a different ecosystem for how we studied every patient. The design overall was for patients with metastatic pancreas cancer, first and second line. The primary endpoint was overall survival. The standard of care arms, both FOLFIRINOX and gemabraxane, were both used, and then we can roll into the trial multiple experimental arms. And an important part is we got our statisticians to be able to design it so that if a patient, the first therapy a patient got didn't work, then they could be eligible for second therapy with both therapies counting for overall survival. So we developed a lot of clinical trial innovations as part of this. Clinical trial concepts were vetted by a committee, and they really could come from any source. We developed an adaptive phase 2/3 platform, a seamless platform. So after safety was shown, a regimen could roll into the platform, which would help accelerate FDA approval of effective therapies, but would also allow us to have fast failure of ineffective therapies. And as an underlay, we built some innovative research, including supportive care, how do we best take care of pain, obstruction, financial modeling to support this, patient-reported outcomes, and all patients got comprehensive genomic analyses and biomarker work. I'm a big believer that you can innovate along the entire spectrum, and there had been some stagnation in how we do clinical trials, so we actually had 12 new clinical trial innovations, many of which never done before in clinical trials embedded in this. This is just a diagram that shows how it works. You have patients across the United States that would be approached to be part of the trial. They were to go comprehensive tumor evaluation, and they would get randomized to either standard therapy, shown here is just a single arm, or a variety of experimental therapies. These, for the phase 2 part of it, it would be no more than 100 patients, and if a therapy hit a certain benchmark, and that being a 65% chance it would work in phase 3, it would roll seamlessly into phase 3. This goes on in time, and as we learn more, we can adjust and add new therapies to the trial. We have had a number of arms roll through. The first one actually was not effective, 50 patients enrolled. We have comprehensive biomarker data, and that's currently being prepared for publication. We have a number of arms that are active and we're awaiting readouts. And then we have a whole portfolio of other arms in development. What's the benefit of this? We have a ready-to-go system, 30 clinical sites now across the United States. This is cheaper and faster to evaluate therapies. This is a registration-ready trial. We had the leadership of the FDA in the United States partner with us to develop it, and it requires fewer patients to get to registration. And interestingly, it's a unique business model to try to do something novel with clinical trials, and I think can serve a model as we move forward for clinical trials, not only in other cancer types, but in also other diseases. In fact, it has been used now for clinical trials for COVID and other cancers. And this is just a map of the centers involved in the United States, and we did purposefully include centers to make sure we could have diversification of patients who could access the trial, and to help underrepresented minorities also have access to the trial. So here are some of the lessons learned, and really it's about trying to do things in a different way, more strategic, to get to effective therapies for patients, and we do expect that we can roll this out for locally confined disease as well. So I showed you this slide at the beginning, and I talked a little bit about a different way to tackle therapeutics, but I will tell you now that I've been operating and taking care of many pancreatic cancer patients over the years, I actually think the most important thing we can do is to pick up this disease early. And you can see the data on the bottom of the slide from a recent paper from the SEER database that shows if you resect someone who has a stage 1A pancreas cancer, their survival rate is 83%. So that's a big difference from 12%. And so we've given some thought and learnings from Precision Prompts to think about how we can do this for early detection. And with that, we conceived the notion of this consortium, which is called the Pre-Seed Consortium, which really started about 5 years ago as just a concept. And what this is a approach to have a collaborative research effort, and now this is over, it's actually up to 55 centers worldwide, where we serve as the academic coordinating center. And the mission is to change the landscape of pancreas cancer by better understanding risk and develop more effective early detection and ultimately prevention tools to get us to a 50% survival rate in 10 years. This has grown to be the largest effort of its kind, and it requires a novel model of data sharing worldwide. So what we have built is a standardized database where clinical, laboratory, and imaging data is put in, and we actually have working groups that have standardized how imaging should be done. We're happy to share that with anybody interested. There are published papers that have come out of this effort, and our goal is to enroll 10,000 individuals at heritable high risk in the first 5 years, and then to perform a 10-year longitudinal study. Centers anywhere in the world can join if they, I'll say, have their act together, which means that they have to have some sort of multidisciplinary team. They have to see a reasonable number of patients, that they have a coordinated effort, and some kind of infrastructure to enter data and collect biosamples. And centers have to agree to share data to join. With such an effort, we can answer a lot of important unanswered questions by standardizing and creating which has now become the world's largest high-risk database. We can generate data that screening saves lives. We haven't proven that yet in pancreas cancer. Only small studies so far show that it may, but we have to really provide convincing evidence. We can optimize evidence-based practice standards. We can study modifiers of risk and develop and really refine our understanding of risk, both for people with familial pancreas cancer, those who carry mutations, et cetera. We all know that about 85% of families that have familial pancreas cancer, we still don't know the genetic cause, and a platform of this size facilitates that kind of research. Importantly, in the last line, with such a large database, we can develop new technologies to develop pancreas cancer in its early stage using blood-based tests, AI, imaging. In fact, we're developing first of its kind pancreatic MRI database, which should allow the application of AI for both early detection and potentially prediction of pancreas cancer. So I very proud of the fact that we opened this study in the middle of a pandemic. The first site opened, first patient in May 2020, and so far, as of last week, we've enrolled over 5,500 patients. We use a nonprofit called Arbor Research that serves as the data coordinating center. They help onboard all the centers. All the centers in the United States have a central IRB. They help all the international centers with their paperwork. We now are moving to a centralized biorepository that has sites across the world. And even though that table on the far right is too small for you to see, it lists all the centers which have a code name and all the different cohorts of individuals at elevated risk on a weekly basis. We get a spreadsheet so we know what's happening worldwide as part of this study. I've actually never seen a study accrue like this, and I think it's something that really resonates with patients. Patients get it. Hey, why don't all you people in academia work together to solve this problem instead of everybody working in a silo? We're currently enrolling about 150 patients a month into the study. You can see on the graph on the right, there's some different lines. We have different cohorts or categories of people who enroll, and we thought it was really important to capture data for anybody who might come into a clinic wondering about their risk of pancreatic cancer. So cohort 1, which is the most common cohort, well over half of the patients are people with familial pancreatic cancer, 2 or more family members with pancreatic cancer, or if they carry a mutation that put them at risk with the presence of a family history. Cohort 2, I just mentioned because this is increasingly coming to our awareness that some of these folks should be screened even without a family history of pancreatic cancer, and those being patients, for example, that are BRCA2 carriers who may have a close to a 6 to 7% lifetime risk, ATM, et cetera. There is heterogeneity in which centers around the world screen those folks, but for all the centers that do, we have full data capture. Cohort 3 is people who should be screened, but they're too young. They're in familial family, and we think they're an important group of patients to do research on, to understand concern about screening and to get engagement. In the United States, there's actually guidelines now that state that every person who has a single first-degree family member with pancreatic cancer should get germline testing, so that represents cohort 4 here for those patients who get screened. We even have a cohort if we want to collect research samples from patients, family members that aren't part of a program, we can collect that. And then cohort 6, which we've been focusing on, are patients who come into our clinics with pancreatic cancer that we find they have familial pancreatic cancer or carry a mutation at the time of diagnosis. Importantly, we want all pancreatic cancer patients to get germline testing, and then we put them in the database and make sure that we do cascade testing on their family members. And we've recently added a pancreatic cyst cohort for centers that would like to use the database for this purpose. The consortium is well organized. We encourage centers that join to have folks at their center join any of these committees. There's no shortage of work to do in this area. And I'll just show you that with zero marketing budget, here are all the centers across the United States that have joined. Every week I get an email, hey, can I join? We want to be part of this. And then here are the centers around the world, and we actually have 2 centers in Australia that are onboarding, but hopefully New Zealand after the meeting. And I think this has been a really special effort to try to tackle early detection in a unique way. We have an amazing body of data that we're generating. Now there are multiple papers coming out. The first one listed there was really a commentary in gastroenterology about us getting our act together as a field to standardize the kind of data we're collecting, but now we're having a portfolio of papers come out, and of course encouraging everyone who participates in the study to tap into the data. There are lots of different scientific projects going on, some of them listed here, and I expect ultimately this should be a project that should generate over 1,000 papers and many, many grants. In fact, any center around the world that is doing any kind of research around early detection can leverage this platform to increase their likelihood of getting funding or publishing papers. It's also been an interesting platform to partner with industry. Our first major industry partnership was with a group of companies. We call it the Lattice Project, where they decided they wanted to elect this as a volunteer project. So people at AWS, Amazon Web Services, this is the good side of Amazon. They basically picked this as a volunteer project to help us organize this at a very large scale. And they gave us a free data cloud for the next 20 years to help us, that we control, to de-identify and load 10,000 patients worth of imaging over a 10 years period, creating the first imaging database for which AI can be applied. And then we now have some companies that will be doing longitudinal testing of promising early detection blood-based biomarkers. These are blood-based biomarkers that look very, very good in distinguishing normal from stage 1 pancreas cancer. We want to see how they perform in a high-risk population. To the point that's been raised in this conference now multiple times, we know that people who not only enter clinical trials, but that are eligible for screening don't always get the screening they need. And so we have established a National Community Advisory Board to help us try to reach into our community in the United States to make sure people at risk, everyone at risk, gets access to this kind of genetic testing and screening. I envision ultimately that all the countries that participate in this would have their own National Community Advisory Board to help execute the same. So in summary, I've showed you in brief 2 platforms, Precision Promise and more recently Precede, that are scaling up. It's time to go big for pancreas cancer and really leveraging an innovative way to data share and empower researchers and clinicians worldwide to lead advances in pancreatic cancer survival. This has been an important learning process that you have to get all the key stakeholders in the room early. And we've had important interactions not only with patients and families as we have always had, but with foundations, pharma, and the FDA to increase the pace and scale, and we can execute change. And I will say, as a active surgeon and scientist, I still get the greatest joy from taking care of patients, and it gives me hope every week when I go to clinic that I'm going to be able to say something new to my patients, that we have new, better drugs and a better way to detect pancreas cancer early. Our goal will really be to increase the use of surgery by finding less than 1 centimeter pancreas cancers, which essentially should be a situation where we should be able to cure most patients. So with that, obviously this kind of effort requires hundreds of people. There you can see a picture of my team in the top left, and but centers all around the world, and really it's really a privilege to be part of the pancreatic cancer community worldwide, which are an amazing group of people to try to tackle this really tough disease. Thank you.
Thank you, Diane. I would really like to applaud the effort which has gone into this, so really well done, and really glad to see some Australian collaboration in that. Perfect. We do have a number of questions, so I'll try to crystallize some of them in one, mostly around Precede. So Diane, where does the funding come from, and in terms of the comprehensive genomic profiling, etc., who's doing that? And yeah.
Precede started by a donation from one of my patients of $1.5 million, where I told them I had this idea. And I actually didn't put that money into our institution. I assembled a committee to find the best data coordinating center, which was Arbor Research, and set it up there as a olive branch of goodwill, and invited centers to join it. And actually, it started by just trying to get all the institutions in New York City to play together in the sandbox, which I guess hadn't really happened when I moved to New York City. And then it just kind of grew and grew. Now there's a nonprofit organization called Trova Now, Find It Early, it's called. That sole purpose is to raise philanthropic funds for Precede. And now people are starting to get grants. We just got a $5 million grant to help support part of it. some of the industry partnerships. So it's really having multiple eggs in our basket to help fund it. We do have some funds so that any new center that joins, we can give them $20,000 a year to help offset the costs for part of a data coordinator. But with some of the biomarker studies we're planning to do, depending on how many patients get enrolled, that could be another $40,000 to $50,000 per center. So we really try to build it so we can democratize the funds across all the centers and empower all the centers to enroll patients.
Yeah. So there's another question which just come through. What have been the barriers to identifying and including new investigative agents on the Precision Promise and the platform when you're interacting with industry?
Yeah, so the question is, how do you figure out what therapies you should put in the platform? So it's really, you know, part of the nice thing about PanCan is they started out funding researchers in pancreatic cancer, I don't know, maybe about 12, 15 years ago, and they helped build a research community. And so really it's identifying interesting potential therapies and new papers that come out. It's at meetings. It could be being approached from a pharma company. So there's a set of guidelines. You know, is there a drug available? Is the science strong? And it gets vetted by a committee that weighs the pros and cons of the value proposition of that arm.
Just had a question for Saxon. So Saxon, how do you determine ethnicity when entered into your system?
Yeah, that's a really good question, and there's been a lot of work to try and improve its accuracy. So it's done through our patient administration system. It's a prioritised ethnicity, and there's been people working back to go back to the GPs to get it right. But essentially every time a patient touches the system, they are asked to just reconfirm what it is, and the accuracy has improved quite a lot over the last few years.
Thank you. A question, just probably last question, one more question, just for Pran. We often talk about centralized and decentralized model of complex abdominal surgeries. Can you comment on that with your practice and experience, and also a comment on post-operative follow-up in regional centers? How do you manage to do that?
Thank you. Our model was actually very similar to Saxon, so our follow-up was very similar. We would keep them close to Townsville post-resection in support and accommodation. Then a lot of the follow-up was done with telehealth. As you know, we also delivered a lot of the all the oncological care locally via our medical oncology team via telehealth as well. So a lot of the follow-up was telehealth. In answer to centralization, naturally, volume is key and to maintaining good outcomes. And early on, we actually centralized all our private patients across to the public system. That way we kept all the cases in one center. And then as we grew and patients' requirements and whatnot, we were able to split and still maintain good volume in both centers.